Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUCET Common Entrance, that is Central University's Common Entrance Test. This was for the MSc Entrance, and this is for the subject Applied Microbiology. It was conducted in the year 10th June 2023. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of this subject or any other subject of CUCET entrance, you can go to my channel Omni Bio Entrance, see for playlist. In the playlist, select for CUCET. In that you will find the question papers and the answers. Then next one. Trichodesmium, a filamentous non-heterocystous cyanobacteria, fixes nitrogen under aerobic conditions in Dead Sea. Yes, these are assertion reason questions. So assertion is correct. Trichodesmium contains a specialized hyaline vegetative cell called diazocyte which protect nitrogenase from oxygen inactivation. That is also correct, and that explains why Trichodesmium, which is a non-heterocystous cyanobacteria, fixes nitrogen in Dead Sea. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then next one. Polysiphonia possesses triphasic life cycle, gametangial phase, carposporophytic phase and tetrasporangial phase. Carposporophyte is a minute microscopic phase. Yes, that's correct. Carposporophyte is found in female gametophytic plant. Yes, that is also correct. Both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then, nitrogen fertilizer is added in rice field in the form of ammonium salt and urea, which is rapidly converted into nitrate. Nitrate is assimilated by plants. However, much proportion is leached, percolated into groundwater or returned to atmosphere in the form of nitrogen gas. Okay, that's correct. Bacteria such as Pseudomonas denitrificans, Paracoccus denitrificans, and Thiobacillus denitrificans are involved in the denitrification process. Okay, so that explains option A why so some of them are returned to atmosphere in the form of nitrogen gas. So the answer here is both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then azolla is a tiny aquatic fern, is used as a biofertilizer in rice cultivation. We already discussed that. Azolla in symbiotic association with Anabena in symbiotic association with azolla. So azolla forms symbiotic association with the nitrogen fixing cyanobacterium Anabena azolae. That's also correct. That is why it is used as a biofertilizer in paddy field. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. Then purine and pyrimidine biosynthesis is critical for all cells. Of course, they are involved in several several factors like DNA synthesis, RNA synthesis, ATP production. All these require purine pyrimidine biosynthesis. Purine and pyrimidine molecules synthesize ATP, several cofactors and other important cell components. Yes. So both A and R are correct, and R is the correct explanation of A. Answer is option 1. The monomers of DNA are called deoxyribonucleotides. The nitrogenous base in DNA is deoxy. A is correct. Monomers of DNA are deoxyribonucleotide. Nitrogenous base in DNA is deoxy. That is not correct. The nitrogenous base in DNA are adenine, guanine, cytosine, thymine. Whereas deoxy, it refers to the sugar component of DNA, that is deoxyribose. So here A is correct but R is not correct. Answer is option 3. Coupled transcription and translation is possible in bacterial and archaeal cell. Of course, they don't have any separate nuclear envelope. In bacterial and archaeal cell, nuclear envelope does not separate the translational machinery from DNA. That is the right reason. So here both the A and R are correct and R is the correct explanation of A. Answer is option 1. Then next one. The dioxic growth curve of E. coli is obtained when grown with a mixture of glucose and lactose. That's correct. The enzyme for lactose catabolism are constitutive. No, they are inducible. When lactose is present, these enzymes are expressed, otherwise they are repressed by the lac repressor protein. So here A is correct but R is not correct. Answer is option 3. Then next one. The transfer of genes from parents to progeny is called vertical gene transfer. That's correct. The genes from one independent bacterial cell can be transferred to another bacterial cell. That statement also is correct, but it doesn't explain the assertion. So here both A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then nitrate is produced during nitrogen cycle. Yes, that is correct. The microorganism use nitrate as a terminal electron acceptor during anaerobic respiration. That statement also is correct, but it doesn't explain the assertion. So both A and R are correct, but R is not the correct explanation of A. Answer is option 2. Next one. Taxol is used in chemotherapy against cancer. Yes. Taxol preferentially kills tumor cells. Yes. That is why it is used in chemotherapy against cancer. So both A and R are correct and R is the correct explanation of A. The next one. Sickle cell anemia disease has the most common form of abnormal hemoglobin. Yes. Sickle shaped. Erythrocytes of the sickle cell anemia patients adopt a sickle shape at low oxygen concentration. That is also correct. That is why it is having a an abnormal shape for the hemoglobin, because erythrocytes of the sickle cell anemia patients adopt sickle shape at low concentration of oxygen. So both A and R are correct and R is the correct explanation of A. Then next one. DNA fragments with sticky ends are particularly useful for recombinant DNA experiments. Of course, sticky ends are more preferred for recombinant DNA experiments. The single-stranded sticky ends can easily pair with any other DNA fragment having complementary sticky ends. Now, if your gene of insert is also having a complementary sticky end, then you can join the gene of insert to the vector DNA, which is also having the complementary sticky ends to the gene of interest, and you can clone them. So both A and R are correct and R is the correct explanation of A. Answer is option 1. Then next one. All eluents for use in HPLC system must be specially purified. Yes, of course, that's correct. The trace of impurities in eluents cannot affect the column and interfere with the detection system. That's wrong. They interfere with the column and interfere with the detection system, such as it can affect column performance, and then these impurities, they can bind to the column stationary phase. Then can alter the pH of the column. Impurities, they can absorb light causing false peaks to appear in the graph. So all these that is impurities can affect the column and interfere with the detection system. So here A is correct but R is not correct. Answer is option 3. Then next one. Type 2 restriction endonucleases recognize and cleave DNA within particular sequence of 4 to 8 nucleotide referred to as palindromic sequences. Yes, that is correct. The palindromic sequence is similar to words that read the same backward and forward. That is also correct. But it doesn't explain the assertion. So both A and R are correct and R is not the correct explanation of A. Answer is option 2. Next one. Silent mutations change the nucleotide sequence of a codon but do not change the amino acid encoded by the codon. That is correct. Silent mutation, they change only the nucleotide sequence, but they do not change the amino acid sequence of the codon. Suppose GUG code for glycine. Suppose. I am not sure about that. It's only an example. Now GUG, the G at the third position get mutated to C. So GUC. Still then it will code for glycine because GUG and GUC, they both code for glycine. So even though there is a mutation in the nucleotide sequence, the amino acid is going to be glycine itself. So silent mutation change the nucleotide sequence of a codon but do not change the amino acid sequence. So that's correct. A single base substitution can change a codon for one amino acid into codon for another. Yes, that is also correct, but it doesn't explain silent mutation. So both A and R are correct, but R is not the correct explanation of A. Answer is option 2. Then next one. Some gram-positive bacteria form endospores. Of course, that's correct. Endospores does not contain dipicolinic acid. No, they contain dipicolinic acid. So R is not correct. So A is correct, but R is not correct. Answer is option 3.